Good morning, respected teachers, batchmates and juniors. Today I am going to present subject seminar on the topic intermediate filaments in health and disease. Here is today's presentation at a glance. We'll deal with a brief introduction, an account on the terminology and key concepts, and then we'll begin to discuss the general features of intermediate filaments, the functions, and then we'll discuss the related diseases and also its role in neoplasia, followed by a brief conclusion and summary. Introduction: the cytoskeleton of a cell consists of three abundant families of fibrillary proteins: microfilaments, microtubules, and intermediate filaments. As we can see in the picture here, three major fibrillary proteins. The first is the actin microfilament, second is the microtubules, and lastly we have the intermediate filaments. This is the one that we are going to discuss in detail today. The human genome consists at least 65 functional genes encoding for intermediate filament proteins, placing them among the 100 largest gene families in humans. More than 30 diseases are related to mutations in these genes. The majority of them are rare or difficult to treat, but collectively they affect most tissues and not all of them are rare, as exemplified by the association of keratin mutations with end-stage liver disease. So before delving into the topic proper, let us finish discussing some key concepts. Intermediate filament proteins derive their name from their diameter, which is intermediate between diameters of microfilaments and microtubules. As you can see here, 7 nanometer, 25 nanometer, and the diameter of the intermediate filament is 8 to 10. That is intermediate between these two, and therefore it gets its name of intermediate filament. The intermediate filaments differ from actin microfilaments and tubulin microtubules in their large numbers, their distribution in the cytoplasm and nucleus, their diverse primary structure, their non-polar architecture, their relative insolubility, and their nucleotide independent dynamics. So now let us go into the general features of all intermediate filaments. Structure: all intermediate filament proteins have a prototypical structure consisting of a coiled alpha helix rod domain. That is, you can see here in this part there is one domain that is in an alpha helix, but then that alpha helix is wrapped on another domain. So it's known as a coiled alpha helix rod domain. That is, two polypeptide alpha helixes wound around each other. This is interrupted by linkers, as you can see here, and they're flanked by the heads, the head and tail domains. The rod domain is consisting of 310 to 352 amino acids. These rod domain parts. Then again the linker parts are coded by 8 to 12 amino acid domains, and the head and tail parts are encoded by 8 to 12 amino acid domains, amino acid units, I'm sorry. Another thing that I would like to talk to you about in this photo is that this head domain consists of units in its head which tell us about the various regulatory changes that the intermediate filament terminal has undergone. For example, all the post-transcriptional and translational changes like phosphorylation, N-acetyl glycolization, transglutamination, all of these changes will be seen by this seen by these specific units in the head domain here. And then here in this tail domain here we can see that it undergoes changes like farnesylation, and also phosphorylation change can also be seen. But also the units in the tail domain interact with apoptosis related proteins, nuclear proteins, chaperones, bundlers, linkers, etc. So you can see how an intermediate filament by the specialized units in its head and tail domain become subject to various regulatory changes. The simplest soluble unit of intermediate filament proteins is a tetramer. consisting of two anti-parallel dimers. Each dimer, in the case, for example, in the case of keratins, consists of one keratin and other type 2 keratin molecule. So basically what they're saying is, first thing is that there is a monomer, this becomes a dimer, but then the two dimers, they will wrap around each other in an anti-parallel fashion. That is, if this dimer will have n terminal c terminal in this fashion, the other dimer will have n terminal c terminal in this fashion. Therefore it will be anti-parallel, it will be this direction to this direction, it will be arranged in anti-parallel form. So the two dimers combine to give the intermediate filament tetramer structure, and polymerization leads to formation of the final intermediate filament. A high-resolution architectural model of intermediate filaments is lacking, although the mature 10 to 12 nanometer fiber is believed to contain 32 monomers in diameter, with important structural polymorphisms among members of the protein family. Now we'll take a look at the classification of intermediate filaments. We see that type 1 consists of acidic keratins. It consists of 28 genes in humans, the details of which I'll be saying in my upcoming slides. Then we have basic keratins, again it has 26 genes. Then we have homodimerizing intermediate filaments, some of which are capable of heterodimerizing, and these includes vimentin, desmin, and GFAP. Then we have intermediate filaments mainly expressed in neurons and muscle. Here we have neurofilaments, nestin, and cinnamon. Then we have nuclear intermediate filaments, which is the lamins family of intermediate filaments. And then we have lens-specific beaded intermediate filaments, which is the facinin and phylensin family of intermediate filament proteins. And here is a much more detailed view. As we can see here, the keratin consists of type 1 and both type 1 and type 2 filaments, and the protein units are K9, K10, 12 to 30, 23 to 28, which are encoded for by particular KRT9, KRT10, KRT12 to 20 gene respectively. Vimentin coded by VIM expressed in the mesenchymal cells, nestin coded by NES gene seen in the neural stem or progenital cells, glial fibrillary acidic protein encoded by GFAP gene seen in the glial cells, so on and so forth. Regulation. Intermediate filament proteins are regulated by several post-translational modifications including farnesylation, phosphorylation, glycosylation, and transglutamination, and also by simple accumulation of associated proteins. So, for example, just if we consider phosphorylation, simply phosphorylation can lead to changes in the solubility, association with interacting proteins, and susceptibility to degradation by apoptosis of an intermediate filament. Only phosphorylation itself can lead to 3 different changes in an intermediate filament. Also, alterations in intermediate filament gene expression is a marker or proof of tissue injury. In addition, there is also transglutamination. Transglutamination of intermediate filaments and their associated proteins under physiologic conditions is essential for the formation of protective cornified cell envelope that contributes to the structure of the skin barrier. In pathologic conditions, however, the transglutamination of intermediate filaments causes formation of a variety of intermediate filament containing inclusion bodies. Intermediate filament proteins are also cleaved by caspases during apoptosis at a conserved motif, and this means whenever there is a mutation at this motif site, that means that intermediate filament which is going to be degraded by apoptosis, that may not get degraded as readily by apoptosis. Why? Because there will be a mutation within this motif. This is a clue in our upcoming part of the seminar, which will lead to discussion on tumorigenesis by intermediate filaments. Let us look at this figure now. Figure here shows the intermediate filament inclusion bodies in various cases. The most common being Mallory bodies, which are seen in alcohol-related disease of the liver. We'll discuss each of these photos one by one. The first photo here is showing specimen of liver with chromoptroph aniline blue stain. We can see here the Mallory bodies, which are indicated by the arrow, and also I'm highlighting them for you. And also the inset picture is showing us a positive immunohistochemistry for keratins 8 and 18, and in all these cases the immunohistochemistry is positive for Mallory bodies. In this immunoelectron micrograph of a Mallory body from the same liver, the double labeled antibodies to keratins 8 and 18 are staining these areas as indicated by the single arrow, and also the antibodies to MM120-1, a non-keratin Mallory body component, is also being shown by the double-headed arrows here. In this photo, that is the hematoxylin eosin section of a specimen of a brainstem from a child with Alexander disease, which is caused by arginine to histidine mutation at position 239 in glial fibrillary acid protein, or GFAP. The arrows here are indicating the extensive characteristic Rosenthal fiber inclusions which are surrounding the blood vessel, and the inset shows micrograph of various fibrous deposits. In this photo, there is a modified trichome stain section of two muscle fibers showing desmin deposits, as we can see here very easily, from a patient with desmin mutation. The inset photograph is showing us electron micrograph of the granulofilamentous deposit. So basically in all these four photos we came to know that whenever there is, due to tissue injury or mutation, whenever there is altered regulation of the intermediate filaments due to phosphorylation or transglutamination, there will always be deposition of the intermediate filaments as inclusion bodies within the tumor, within the cells. Within the cells. So this panel here is showing us once again double stained specimens of cirrhotic liver, and the inset showing us normal liver. The first photo is showing us positive vimenting staining denoted by the green areas, and we can see that in the inset in the normal liver there is no green area. That means in the cirrhotic liver there are the larger image is showing us a fibrotic liver with positive vimentin staining denoted by the green areas. The second photo, again we can see in the inset there is a normal liver, and it is only having red marks. That is, it is stained only by antibodies against keratins 8 and 18. However, in the larger picture we are also seeing some yellowish areas, because that is being also stained by keratin 8 apoptotic fragment, and therefore it is appearing yellow. Panel C shows a transmission electron micrograph of a basal epidermal keratinocyte from a patient with an arginine to cysteine mutation at position 125 in keratin 14, which caused Dowling-Meara epidermolysis bullosa simplex, the most severe form of the disease. The aggregates of keratin proteins that are not seen in normal keratinocytes are present here, as we can see here. But also this cell is also showing us an unstressed situation, because it is containing the normal appearing keratin filaments also, as we can see here. We'll continue the presentation with the discussion on the functions of intermediate filaments. Shared and tissue-specific functions. Difficulties in unraveling the functions of intermediate filament proteins are related to their poor solubility, the lack of specific pharmacologic agents to inhibit them, their apparent absence in genetically tractable organisms such as yeast, and the lack of an essential biological function when they are tested in culture systems. The best understood function of the intermediate filaments is that of a scaffold for maintaining cell and tissue integrity. The diseases that are related to intermediate filaments and that manifest as tissue, cell, or nuclear fragility in the skin, cornea, or muscle, and some of the laminopathies support this view. In addition to scaffolding, intermediate filament proteins protect against non-mechanical stress and decrease susceptibility to apoptosis. Other roles of intermediate filaments include tissue-specific functions such as those of neurofilaments in dendritic arborization and in radial growth of myelinated axons. So disease-related aspects also we'll see. An important aspect of the genes encoding intermediate filaments is the selective expression in certain cell types during differentiation. This feature is relevant to the association of mutations in these genes with a broad range of tissue-specific diseases. The clinical manifestations of abnormal intermediate filament proteins can be due to mutations with high penetrance, such as those that cause epidermolysis bullosa simplex, also mutations predisposing to disease, such as end-stage liver disease, which may be associated with mutations in the genes encoding keratinins 8 and 18, as we have already discussed. Neurofilament mutations, which may predispose to amyotrophic lateral sclerosis and aberrant accumulation of intermediate filament-rich inclusions or aggregates, such as Mallory bodies in liver diseases and Rosenthal fibers in Alexander disease. Furthermore, intermediate filament proteins can serve as markers of the tissue origin of poorly differentiated tumors. Keratins define epithelial tissues, whereas vimentin defines mesenchymal origin as tumor markers in serum and as a means of detecting micrometastasis. So now that we have finished discussing the functions and the disease related aspects of these functions, we'll look at the diseases proper that are related to intermediate filaments. Diseases of the skin and epithelium. Ultrastructural studies performed in the 1980s showed that epidermal basal cells from patients with epidermolysis bullosa simplex contain dense cytoplasmic aggregates. In the late 1980s and early 1990s, various experiments showed that keratin mutations cause keratin aggregation in cultured cells and mechanical fragility of epithelial cells in vivo. Soon thereafter, mutations in the genes encoding keratins 5 and 14 were described in patients with epidermolysis bullosa simplex, which is associated with blistering in the epidermal basal cell compartment, where keratins 5 and 14 are the major keratins. After mild frictional trauma such as rubbing of the skin. In this panel of photographs, we can see that patients with intermediate filament related disorders, the intermediate filament proteins that when mutated cause the disorder are also indicated in this photo. For example, the first photo we can see keratins 5 and 14 and they are causing epidermolysis bullosa, a particular type of epidermolysis bullosa. We'll discuss details of each of these photos in the upcoming slides. So the first photo, it is showing us the Dowling-Meara form of epidermolysis bullosa simplex. It's caused by heterozygous arginine to cysteine mutation at position 125 in keratin. Blisters occur because of structural weakening of the epidermal basal cells where keratins 5 and 14 are predominantly expressed. The second photo is showing us mutation in keratins 1 and 10 causing epidermolytic hyperkeratosis, causing widespread hyperkeratosis as we can see in this photograph, along with skin fragility. This last photo is a slit lamp image in which microcysts denoted by the arrow are seen in the corneal epithelium. This is from a severely affected patient with a keratin 12 mutation. The intermediate filament cytoskeleton of the anterior corneal epithelium consists of largely of keratin 3 and 12, and the mutations in this keratin cause Meesmann corneal dystrophy, which is characterized by microcysts, corneal microcysts due to cell fragility. It can be asymptomatic in some cases, but in others it leads to irritation, photophobia, sensation of foreign body, and in few cases corneal scarring and loss of visual acuity. Dominant mutations in the genes encoding keratin 1 and 10 and 1 and its partner keratin 10, which are expressed in the supravasal epidermis, were associated with epidermolytic hyperkeratosis. These studies established that keratin diseases are characterized largely by fragility of the affected tissue and that in some cases they are accompanied by hyperplasia of the tissues expressing the mutant keratins. A broad range of skin and other epithelial diseases have been associated with keratin mutations. In general, these disorders are caused by dominant negative mutations, most of which are missense or small inflame insertion or deletion mutations, primarily affecting the keratin rod domain. Mutations affecting the helix boundary motifs of the rod domain generally cause severe disease and have high penetrance. Mutations occurring elsewhere in the gene usually cause less severe disease or lead to disease susceptibility, as seen in mutations in the genes encoding neurofilaments keratins 8 and 18. Less disruptive mutations can also lead to a clinically distinct skin disease affecting only pressure-exposed palm and sole epidermis, as in epidermolytic hyperkeratosis. Mutations in the keratins 8 and 18 genes do not disrupt filament formation and have been identified as risk factor for hepatic cirrhosis. Diseases of the hair. Keratins are also subdivided into soft keratins, that is epithelial and organ keratins, and hard keratins, that is trichocyte keratins, or specialized epithelial cells that terminally differentiate to form hair and nail. Monilethrics is a keratin-related hair disorder characterized by beading and fragility of the hair. which is caused by mutations in HB1 and HB6, the genes encoding human hair keratins HB1 and HB6. Several other epithelial keratins are expressed in specific hair follicle regions. Of these, sequence variants in keratin 6HF are implicated as risk factor in the loose anagen syndrome and pseudofolliculitis barbae. The overlapping distribution of keratins in hair follicles probably accounts for the relative paucity of known intermediate filament disorders primarily affecting the hair. However, it is likely that keratin genes, because of their multitude and their compartmentalization within hair substructures, play a part in other hair disorders and in normal variations in hair characteristics. Myopathies and lens disorders. In Desmin myopathy, identified in 1998 as the first non-keratin intermediate filament disease, this muscle disease is similar to that in epidermolysis bullosa simplex with muscular dystrophy. Gain of function mutations in alpha B crystallin also cause Desmin related myopathy, whereas loss of function mutations cause congenital cataract. Alpha B crystallin is a chaperone that regulates the organization of several cytoplasmic intermediate filaments, including desmin in muscle and facinin, and filancin in the eye. Similarly, mutations in facinin underlie autosomal dominant cataract, and it is likely that mutations in filancin, its partner protein, cause an analogous disease. We also have neurodegenerative disorders. Mutations in neuronal intermediate filament proteins are related to several neurologic diseases. For example, Alexander disease, a neurodegenerative disorder, and the mutations in neurofilament light chains in axonal Charcot-Marie tooth disease is also an example, and mutations in neurofilament heavy chains and periphery as peripherin 66 as potential causes of sporadic amyotrophic lateral sclerosis is also there. Interacting protein related diseases. Proteins that interact with intermediate filaments in desmosome and hemidesmosome cell junction complexes are also associated with several diseases. The loss of plectin, a cytoskeletal linker protein with critical functions in keratin organization in skin and desmin organization in muscle sarcomeres, causes the recessive disorder epidermolysis bullosa simplex with muscular dystrophy. Disturbances of other desmosomal proteins, that is plakophilin, plakoglobin, desmoplakin, desmogleins, and corneodesmosin, have been linked to ectodermal dysplasia, hair abnormalities, and cardiomyopathy. Lastly, we'll discuss the pathogenesis of cytoplasmic intermediate filament related diseases. We can see here that in the cytoplasmic intermediate filament mutations, there is fragility of the cell, there is cellular stress leading to apoptosis, and also altered regulation and dynamics in the cellular cytoplasm. Whereas in the nuclear intermediate filament mutations, we can see that there is fragility of the nucleus, endoplasmic reticulum dysfunction is there, and also altered gene expression, and also it leads to cellular stress and apoptosis. In the end result is in both cases there is fragility, and in both cases they lead to either minimal disease, either enhanced disease susceptibility, or alternative diseases. Next we have laminopathies. Mutations in LMNA gene, which encodes the developmentally regulated lamin A and C isoforms, have been implicated in 10 disorders. These conditions are generally late onset diseases that affect differentiated tissues in a highly selective fashion, as in dilated cardiomyopathies with conduction defects, or in a more systemic fashion, as in premature aging syndromes. Several of these disorders were reproduced in mice with null or point mutated LMNA alleles. Like mutations associated with cytoplasmic intermediate filament related disorders, the mutations in non-lamin proteins can cause diseases that mimic laminopathies. For example, mutations in peroxisome proliferator activated receptor gamma cause familial partial lipodystrophy. Multiple lines of experimentation have established that the nuclear lamina participates in determining the shape, size, and integrity of the nucleus, the number and positioning of nuclear pores, and the organization of transcriptionally silent chromatin. Owing in part to the timing of their appearance and pathologic features, the muscular lesions elicited by lamin A and C mutations are consistent with defects translating into loss of nuclear integrity after repeated trauma. Yet most of the tissues affected in laminopathies are not exposed to substantial trauma, and unlike cytoplasmic intermediate filament disorders, a large fraction of the disease-causing mutations are located within the non-helical tail domains of lamins A and C. Pathogenesis of laminopathies. The pathogenesis of laminopathies is probably related to other lamin functions that are just beginning to be understood, including the control of gene expression and effects on the endoplasmic reticulum, which is contiguous with the nuclear envelope. The first function is supported by the binding of lamin to chromatin, specific transcription factors, and proteins associated with nuclear processes such as DNA replication and RNA processing. The second possible function stems from the demonstration that key nuclear envelope components are aberrantly distributed to endoplasmic reticulum cisternae in the fibroblast of patients with null or mutant lamins A and C. How the function of the endoplasmic reticulum may be disrupted by these mutations is not understood. Such disruption can either elicit a general cellular stress response or alter the cellular response to specific stresses such as heat shock. Additional studies are needed to ascertain the mechanism underlying the variable effect of lamin mutations. In this regard, the atomic structure of the lamin A tail domain reveals that mutations in lamin A and C cause familial partial lipodystrophy and Emery-Dreifuss muscular dystrophy, and may act by disrupting interactions with key associated proteins. And here is a summary of all the entities that we have discussed till now. We are the table begins with the keratin related disorders. As we can see here, for each and every disease that is discussed here, chronic pancreatitis or cirrhosis hepatitis, the gene that is involved, the mutated gene that is involved, is also indicated. For all of these, we can see here. For epidermolysis bullosa, we can see keratins 5 and 14. For epidermolytic hyperkeratosis, we can see keratins 1 and 10. We can also see for loose anagen syndrome, we can see keratin 6HF has been implicated, as we have already discussed previously. Also, we have discussed pseudofolliculitis Barbe. Again, keratin 6HF mutation is implicated. Same for the laminopathies. We can see that the Charcot-Marie-Tooth disease has been implicated. In these, lamins A and C have been implicated. In fact, in all of these laminopathies, lamins A and C are the only lamins that are implicated. So now we'll discuss role of intermediate filaments in neoplasia, that is tumorigenesis, metastasis, and also diagnosis. When normal cells transform to cancerous cells, original signature expressions of intermediate filament proteins are largely retained. As the vast majority of cancer cells originate from epithelial cells, staining of intermediate filaments, most notably keratins, has proven to be a useful tool for pathologists to identify tumor and cell types. Expression of keratins can also be used to classify cancer types. For example, in non-small cell lung cancer, K17 is overexpressed in squamous carcinomas compared to the adenocarcinomas, whereas the reverse is true for K8 and K18. A growing number of studies are showing that altered expression of a subset of intermediate filament proteins might contribute to patient prognosis. This is most notable for vimentin, which is upregulated in cancer cells that have undergone epithelial to mesenchymal transition. Originally discovered in neuroepithelial stem cells, nestin is a class of type 6 intermediate filament protein that continues to be used as a marker of neural stems and progenitor cells in the central nervous system, but is also expressed in other tissue types, including endothelial cells. With a long C-terminal, and nestin is a large protein at more than 170 kDa that cannot form filaments by itself, but instead interacts with other intermediate filament proteins to form filaments. In cancer, nestin has been found to be expressed in cancer stem-like cells and poorly differentiated cancer cells. Mechanisms of change in intermediate filament gene expression in cancer settings involve transcriptional regulation, but recent findings also suggest that post-transcriptional as well as post-translational regulations are also involved. So with all that background knowledge in mind, let us look at a summary of like picture here. which is telling us what are the various levels at which intermediate filaments regulate the various signaling pathways, and therefore what are the various ways where mutation of such a intermediate filament can lead to alteration of the stimulation of that pathway, for example either upregulation or downregulation of that pathway, and therefore leading to promotion of one of the cancer hallmarks and therefore leading to tumorigenesis. For example, let's consider K19 here. K19, we can see it is involved in the SRC, the HER2, and the Notch signaling pathway, and all of these have pro-tumorigenic effect of proliferation. That is one of the cancer hallmarks. Again, we can see that K23, K19, K80, K8, and K14. All of them, though they are affecting different signaling pathways, all of them lead to migration and invasion hallmark of cancer. So, and again we can see nestin here. We can see that by inhibiting CDK5 it controls cell stemness, but due to mutations or due to tissue injury, whenever there is alteration of this intermediate filament protein expression, this will lead to alteration of the signaling pathway and therefore lead to one or more hallmarks of the cancer being propagated and therefore lead to tumorigenesis or tumor invasion and metastasis. So, activating invasion and metastasis. The requirement of intermediate filaments on metastasis occurs at two levels, which are not mutually exclusive. First, as polymerized filaments, the intermediate filament proteins provide mechanical element that enables cells to invade and migrate through their surrounding tissues during initial stages of tumor metastasis. This element of intermediate filament proteins is likely to involve other cytoskeletal proteins, as mechanical properties and dynamics of intermediate filaments are linked to those of microtubules and actin filaments to propel cells forward. Secondly, intermediate filament proteins have been found to regulate signaling pathways in cell migration through their interacting partners. Among intermediate filaments, vimentin and nestin are positive regulators of cell migration and invasion, due in part to their roles in signaling events during epithelial to mesenchymal transition and stem cell maintenance. The last topic for today's presentation is recent advances. Given the clinical correlation and the role of intermediate filament proteins in tumor progression, therapeutic regimens against intermediate filaments, and therapeutic strategies against its associated signaling network may benefit cancer patients. Indeed, withafferin A naturally derived bioactive compound that targets vimentin and induces vimentin cleavage, suppressed tumor growth and metastasis in various mouse models. However, withafferin A is the only small molecule that inhibits intermediate filaments structure and function, and it targets several other cellular components also. Thus, it will be important to develop intermediate filaments protein-specific small molecules to modulate their function in cancer. Developing such a region, along with identification of the mechanism underlying intermediate filament mediated tumor progression, will be invaluable for cancer patients clinically. So the conclusion for today's presentation is intermediate filament proteins make up the largest family of cytoskeletal proteins and are traditionally known for their roles in fostering structural integrity. Intermediate filaments play key roles in various functions of our body. For instance, pro-tumorigenic intermediate filament proteins, vimentin, nestin, and a subset of keratins, including K17, even if they differ in the expression pattern, share a common trait in that all these proteins become induced upon wounding and tissue injury when cells undergo hyperproliferation. However, several intermediate filaments proteins remain unassociated with human disease. Example, lamins B1 and B2, vimentin, keratins 7, 9, and 20, alpha-internexin, syncoilin, and cynamin. But it remains highly possible that mutations in the genes encoding some or all of these proteins will be found to be related to certain human diseases. In cancer, intermediate filament proteins serve as a diagnostic marker as tumor cells partially retain the original signature expression of intermediate filament proteins. They also play a key role in tumorigenesis and metastasis. Going forward, it will be important to determine how different intermediate filament proteins present in a given tumor coordinate their functions to affect tumor progression. Not only are several keratins present in the same tumors, but in certain cancer types, vimentin is co-expressed with keratins. The summary: Intermediate filament proteins take on an active role in cancer development, progression, and metastasis. Despite differences in expression pattern and some of the cellular functions, there are many similarities between intermediate filament proteins, and the knowledge gained from one intermediate filament protein could very well be applied to others. Mutations of protein expression of intermediate filaments are the root cause of all intermediate filament associated diseases. Intermediate filaments, by their altered expression, can induce tumorigenesis and metastasis, but also due to the retention in the tumor cells, are the primary target for diagnostic and therapeutic modalities. These are my references. Thank you.